Good morning, everyone, or good afternoon, depending on from where you're joining. I am Martinez, a product manager in Stevanato Group, and I'm going to be talking to you about the manufacturing of a drug containment solution, quality by design. So we're going to cover three main topics during the presentation. We're going to start with the different container types, their challenges, and a quick overview of their critical quality attributes, to then deep dive into the vial and syringe manufacturing process. So let's start with which are the main challenges of pharmaceutical glass packaging. And from here, I would like to start with the four main containers used in the market. We're talking about prefillable syringes, cartridges, vials, and ampoules. Each of these containers target different therapeutic areas or different applications. We see on the prefilled syringes, mostly used in biologics, anticoagulants, vaccines, some cosmetic applications like hyaluronic acid, and vial use in autoinjectors. While for cartridges, it's more focused on diabetics, dental anesthetics, hormones, or emerging drugs in combination with an autoinjector as well. But when we're talking about vials, the application is broader. We have biologics as well, we have vaccines, we have drugs for IV infusion, generics, and a general use overall of vials in the pharma world. Ampoules, I'm not going to go into the detail of them. We know that they're mostly used for generics, small molecules, or diluents as well. An important thing to mention about the different containers is how they are sealed, how they're closed in order to maintain the CCI. Each of these containers have different materials and different applications, so of course their closures are also different. In prefillable syringes, we see a rubber plunger on the back end of the syringe and then another rubber closure on the tip end of the syringe. It could be a needle shield, rigid needle shield, or a tip cap, depending on the format. On the cartridges, we have always a rubber plunger on the inside, but on the tip side, we don't have a needle shield. We have a lined cap, which is a rubber liner with an aluminum that creates the seal. When we're talking about the vials, though, the vials have a different system, which is the rubber stopper plus an aluminum cap, or lastly, we've seen a lot of introduction of press feed caps into the market. So we need to take these different sealing types in consideration when we're talking about the challenges of each container and what are the critical attributes. So let's start with the vials. We mentioned here a couple of the critical quality attributes of vials, just to make sure we cover the basics. We have the mechanical attributes, which are the mechanical resistance to avoid vial breakage. We have the type of glass. There are different types of glass, so we need to take that into consideration, or additional treatments to the outer surface. Well, functionally, the vials need to be compatible with that downstream process. So fill and finish operations, labeling, even vial adapters for using the drug itself. There's also the cosmetic aspect. We want to avoid false rejects or glass flaws that could compromise the inspection of such drug product. Then we have the actual functional or the closure to maintain the integrity of the drug product. So we're talking about CCI, different rubber types, different geometries, and of course, the different designation of what's going to be. Is it going to be a live process? Is it liquid filling? So we have to take all those things into consideration. Then regarding the glass itself, we have the chemical aspect. So what's going to be interacting with the drug product? Normally, we're talking about the alkali release, the lamination propensity, or any other additional internal treatments that a vial may have. Switching towards the syringes, we have also the critical quality attributes, but we're talking about slightly different things. It's not only the closure, so the CCI, where we have, again, the different rubber types, the geometries, the functional aspect of how you open the syringe, but we add different aspects like the functionality. We need to consider the silicone quantity. How is the coating performing stability-wise throughout the shelf life of the product? What's the delivered dose? What's the dead volume? We have the mechanical aspect as well, where we see the mechanical resistance, functional performance, and dimensional compatibility with dimensional outreach. Always talking about the functionality of a syringe, we need to be careful of the gliding forces, so breakthroughs and gliding forces. What's the viscosity of the liquid looking like? This will have a direct impact. And again, the silicone stability will have an impact also through the shelf life of the product. At the end of the syringe, we have the plunger, which could also have an impact on the CCI. And again, we have the different plunger types, geometries. We have the air gap, the fill and finish process, and the interaction that this will cause with the product. So overall, these are some basic critical to quality attributes we need to take into consideration when manufacturing these containers. So a summary table of these challenges is represented in this chart, where we see the different factors that could cause different challenges to the drug product and then where the containers stand. You see not all factors are similar or are cross-board with different containers, but some of them are, like the ENL, so extractables and leachables. So what are the materials that are in contact with the drug product? We have several that are dedicated to some containers, like tungsten residues that are dedicated or only present within a syringe system. As the other containers, during the manufacturing process, there is no tungsten. So there is no potential of contamination of tungsten residues in the container. Said this, I would like to start a little bit more into the manufacturing process in order to mitigate these risks and try to build the best container for the drug product from the start. So that's why we're here. So I'm going to talk you through the manufacturing process of a vial, and this is our standard layout of the line. So you will see here numbered different aspects of the line, where we start with a glass tubing loader, where it takes the glass, puts it into the vertical warehouse, into the glass forming line. During the glass forming line, we're going to shape the vial, form the bottom, and inspect it. Once it's inspected, it will go into the after forming line, which has several purposes. The first one is to cool down the vial, and the second one is to do additional controls or additional treatments. Then it will go into the annealing layer. The annealing layer has the still purpose of releasing the tensions created to the glass, that were created during the forming process. Once we exit the layer, we're into a clean room where the cosmetic inspection will occur, and then a final packaging in case it's a bulk vial, or it will get transferred into the sterile process if it's a ready to use, so ready to fill vial. So I would like to take you to the next slide, which is the video of how this forming line is actually built. So here we see the first step is the rotation forming machine, where we'll be able to see the forming of the bottom. Here's where we distribute the glass and create the bottom. You see the forming of the neck, so the finish of the vial, where you see the two rollers coming in, the plug in order to give it the internal diameter. This is created by a set of different processes. First, you heat up the vial, and then you shape it until you give it total length, and then pass it on to the after forming line that you see here. So as I mentioned, these vials are controlled both during the forming process and after in the forming line. Here we are performing a total length and cavity control, as well as the control for the inner dimension of the bore of the vial. After here, you see here, there's a possibility to do additional treatments. Here is based on customer request of the product itself, if it requires a treatment or not. And then from there, the vials will get transferred towards the annealing oven, where we're going to release the stress. These vials, though, as you can see throughout the process, have no glass-to-glass contact. This is in order to maintain the quality that was created and avoid defects downstream. Here is the ingress into the layer. The lever has a controlled temperature curve, where we are going to be releasing the stress of the containers. Once the lever goes into the cosmetic controls that we saw inside. So we here see how the controls are performed. So I said we control 100% of the critical dimensions in order to ensure a compatibility of the vial from the dimensional side. And as I was saying, into the room, there is a cosmetic control where we are going to try to reject those vials that have dimensions that are outside of the AQL foreseen with the customer. So as you saw during the manufacturing process, there are different aspects that we can control. And we need to be able to guarantee that this is met in order to guarantee the compatibility with the drug product. So a quick example of this, I did two examples here. One is the lamination factor, that some of the causes of the lamination are coming from the vial manufacturing process. So in case the drug product is susceptible or is prone to interact with glass, we need to make sure that the forming process is the adequate or the optimized forming process in order to guarantee the compatibility and that the lamination will not occur. The other aspect that I mentioned here is also the lyophilization, for example. A vial for lyophilization needs to have certain considerations, both in the vial itself or also in the primary packaging, which will be your secondary packaging. So all these different aspects needs to be designed from the start in order to guarantee a successful launch and a successful vial or a successful product. Here again, it's just two examples of two different things that can be optimized from the forming process in order to avoid problems downstream. So if we go now towards the syringe manufacturing process, you will see that it's quite similar to the vial manufacturing process, but it has some differences. So currently syringes are widely used only in RTU format. So we will go through the converting process again, as we did for the vials, but then we will also go through the sterile process. So where we wash, siliconize, and sterilize the syringes in order to brand them to our customers ready. Here we have the typical line, the typical layout of a syringe line. And you can see it's very similar to that of glass. That's because the process of forming a glass syringe or a glass vial are pretty much the same, except for when we're cutting the container from the glass cane. So we will see again that we have the first step is always the glass tube loader. So we will take the glass and load it into the vertical forming machine. We then have the forming of the syringe itself. And again, we will have, because the camera control for all the dimensions regarding the cone of the syringe. We will then pass towards the after forming line. A difference between the vial and the syringe after forming line is that in the after forming line of syringes, we do not perform additional treatments, but we do perform additional controls, like total height or the inner diameters and flange diameters. In the after forming line, we still have the annealing layer, always to release the stress created during the forming process. And then once we exit the annealing layer, we go always into a clean room ISO 8, where we do the cosmetic controls and the packaging. In case the syringe is a stake needle syringe, we'll then go into the needle assembly line before moving it towards the sterile process. Important to mention as well as for the vials, we have all process. There are no glass to glass. We found new materials for the annealing layer where we are actually eliminating the glass to metal contact in order to reduce again defects that could potentially be introduced into the container. And we have an optimized thermal forming cycle in order to avoid stressing too much the glass. So what we'll see here on the left side is the forming of the glass syringe. We'll form the shoulder, heat up the glass, start forming the cone, heat up the glass, and do the final forming. It's very similar to that of the vials. The main difference is that the pin that goes into the core is actually made of tungsten, and this is where the tungsten residues come from. Once the syringe is cut, we form the flange. Again, we heat up the glass, form the flange, heat up the glass, cut it, and then there is an inspection. The inspection, in this case, is for the thickness of the flange and the flatness. But as I was saying before, and the after forming line, we will perform additional controls where we are controlling the total length of the syringe as well as the diameters and outbound of the flange, both the outside diameter and the inside diameter. Here in the after forming line, again, in the syringe, we are not performing additional treatments. We are only performing the measurement controls. The syringe will get placed into the annealing oven where we're going to be doing the relief of the tensions formed during the forming process. I'm going to skip the needle assembly part and go directly into the seal manufacturing. So now we are heading towards sterile production of the syringes. As you can see here, the syringes are coming from the bulk forming line. They're being transferred in these plateaus that we'll see in the video in the next slide. And that's going to be the in-feeding of our system. Once we are inside the system of these fill lines, we will perform washing and siliconization. We'll perform the capping of the syringe. We'll put it in the nest, seal it, print it, bag it, and then finally send out to refill. During all this process, we will perform additional controls to make sure that what we are introducing into a syringe is according to specification, as well as we are not creating additional defects because of the handling of the syringe. So, for example, after we assemble the closure, we will make sure that the closure is positioned correctly, but we will also make sure that the flange was not damaged. Since the syringe needs to be held from the bottom in order to assemble the closure, there is pressure that's being applied to the syringe, so we need to make sure that we've not introduced an additional defect on these syringes. So let's move to the next slide so we can see here the video of how the syringes are processed. So as we discussed before, the ingress into a plateau in order to avoid syringe handling is automatically done by the robots, where they check the presence of the syringe in the orientation. The robot will take it into the washing machine, where we have the classical 3 washing, the final step with WFI, drying with pure filtered air, and then we go into the siliconization. What we see here is the siliconization of the needle. There is also the siliconization going up the syringe to typing muscle technology, and then we check if there is any clogged needles in the case. We pre-position the closure, in this case is an RNS, then the closure will be finally pressed into a syringe to its final position, where we, after this step, as I was mentioning before, we will control that there was no damage done to the flange. Once the syringes are compliant and siliconization presence and distribution is verified by camera, still go also as well for the vent closure control to make sure the assembly was done correctly. And in case necessary, we will also have the availability to do an X-ray control. If syringes are good, then they will be nested, as we see here, and then they will be inserted in the tub and go to the packaging for then final sterilization. All the sterilization of the syringes is performed by ETO, as heat sterilization is not inappropriate, given that they will damage or it will reduce the functionality of the glue in case of a stake needle. So what I want to discuss and what I want to say is that all glass manufacturing or all the container manufacturing companies, being Stevanato Group or any other, are usually asking a lot of questions to pharma companies. The main reason that we ask all these questions and that we need to share the information is because all the downstream processes that happen to our containers, so from if it's a bulk container, the washing, depergenization, filling, capping, and then storage of the container will have an impact on how this is behaving in the product. So as we discussed and saw throughout this presentation, there are many variables to be taken into account in order to properly design and manufacture a glass primary packaging container. So some of these examples is the drug product formulation, the raw material that is being used for the container, both on the glass and on stoppers, the type of packaging. So is it a vial, is it a syringe, is it a cartridge, how this packaging is being used, is it going into an auto-injector or not, is it going, if it's a vial, is it going to a lyophilization, is it liquid, is it a powder? So all these different processes and aspects of the downstream of a manufacture of a vial or a syringe has an impact and needs to be taken into consideration when we are designing before manufacturing. If we don't have the vision as to what the container is going through, then we might provide a container that is not adequate or that will not perform as we intend to perform or as you are expecting to perform. So this is why we ask a lot of questions. This is why we need to work in partnership with our customers in order to avoid potential delay or potential problem downstream in the life cycle of the drug product. Thank you.